So I will be talking about the key concepts in establishing a metabolomics study and how to design and complete a successful experiment. So what ultimately is our goal of metabolomics? We have a list of metabolites, but what do we do with those? So we can go any number of ways, whether we're interested in what kinds of pathways are perturbed, or whether we want to do some type of model construction to figure out a mechanism or develop some type of hypothesis. But a lot of times, especially for clinicians, they may be interested in biomarker discovery. And so let's say we have some type of disease versus healthy control where we have some type of fold change. We want to go on and validate those either using targeted mass spectrometry or if we want to use some other type of parallel platform. So the typical metabolomics workflow, you theoretically hope that you have a hypothesis that you want to test. But the important part is the experimental design, because if you don't actually have a good experiment, then again, junk in, junk out. You want to think about the storage and stability as well as the extraction procedure that you're using. And not only that, the analytical method, and then Dan and Ursa will talk about the data collection and the statistical analysis aspect of it. And you want to go on to do some type of pathway analysis, and this is the good part about it, the subsequent experiment that you want to go on to afterwards. So essentially most of us start with a small pilot study, and then we go on and from those results we can go in and do a large, increase our N, or power. So here are some of the experimental decisions that you really want to consider. In terms of the study design, ultimately you want to think about your population, the race, gender, age, the study type, whether it's population-based or if it's clinical studies or animal or cell models. And the sample type, it's not just limited to blood or urine or saliva. You really want to think of whether you're doing plant metabolomics or, pick a sample that's representative of the question that you're asking. You also want to think about your sample collection. And so this is actually quite important, and my colleague will show some case studies for the second half of this, which really, you can really see where, that makes or breaks your analysis. Collection time is also important, so you have to think about whether you want all your subjects to be fasting or non-fasting, or if you want control if you want to control for the nutritional challenges, but also sample quantity. And so a lot of the issues is, you collect your samples in one tube and then you want to rerun those for another experiment. You keep going through freeze thaw. So you want to make sure that you have multiple aliquots to ensure that you want to avoid freeze thawing. Sample storage is also important. The temperature, whether you want to use negative 80 degrees Celsius or if you want to use liquid nitrogen. But also aliquoting. So again, you want to make sure you have the right amounts of sample for the platform that you're using. So 1 ml example for NMR, or if you want 200 microliters for mass spectrometry. Again, it depends on the sample, the biofluid that you're using. So whether you think that you'll need more for urine or less or more for plasma. So that's again based on your sample design. And biobanking again, so I know a lot of us get samples from biobanks, and that's actually important because we want to make sure that we know how those samples were collected, how long they were stored for. Sample preparation is also quite important. So whether you have to do some type of metabolite extraction or if you know exactly what you're looking for, then you can then do a specific extraction for those, whether it's solid phase extraction or if you're doing NMR or if you're doing GC, and if you need to do some type of derivatization to figure out what it is that you're, to detect what you're interested in. And so that brings in the platform. There's obviously NMR, there's LCMS, there's GCMS. And so you have a wide range of different options in terms of whether you want to do targeted analysis or non-targeted if you have no idea what you're expecting to find out. And so you want to do some type of non-targeted to really give you an idea now of what to go in and later further target. And again, you can do semi-quantitative as well as quantitative analysis. The data analysis aspect, I'll leave that to my colleagues to discuss later on, but again, that's something very important, and you want to choose the right type of analysis for the questions that you're asking. So again, that brings us to the basic steps to experimental design. So really you want to have your experiment as controlled as possible so that you can either test the hypothesis or generate a hypothesis. So obviously if your experiment's not very controlled, then you don't have much faith in the results that you're getting. So usually the person doing the experiments are either interested in either the effect of certain diseases or the effect of treatment, or if you're not clinical, if you're, plant metabolomics or another aspect, you may be interested in, the effect due to something. So this brings us to how much is enough, and that brings us to the power. And so for the sample power, obviously the greater number of samples that you have, the greater N that you have, then the more power you may potentially have in your study. But again, you have to think about what's feasible. So if, for example, you need 2,000 samples to do the analysis, but you only have a few $100, then obviously that's not feasible. So you kind of have to weigh in on how many samples do I need, and then how much money do I have to actually do this, because obviously the more samples, the more money. Ideally, in a perfect world, if we had hundreds of thousands of samples to test our hypothesis and say, oh, this is great, but we know in reality that that's obviously not possible. But again, it's essential to really plan and design an experiment prior to, so that you can get the best bang for your buck. And so, for example, and we'll bring back the power analysis for metabolomics. It's typically conducted using known values and variation for a single molecule. So, for example, let's say we have a certain disease, and we know that testosterone may be a player in this. And we know that it ranges from 80 to 100 milligrams per liter in men with a 10% variation. We have to figure out how many samples we need to get a certain percent of power to determine that difference. But the issue with metabolomics is that we're measuring thousands of molecules. So how do we get that type of power? Well, you have one of two ways you can go about this. You can either calculate your average variation, so you look at all your metabolites across the board and across all your different sample groups and figure out what that variation is. Or you can use a single molecule as an example. So for example, if we know that we're only interested in the amino acids, maybe we look at one of the amino acids. Again, this is really based on the question that you're asking, the study, the population that you're looking at. But there's a lot of tools out there to help you determine the power prior to your analysis. And so there's tools, there's G-Power, for example, and for the people familiar with R, there's different software packages that can really help you determine that power prior to. And if you already did an analysis, you can use those to figure out what that power is for your study. And then if you had a pilot experiment, you can use those preliminary results to figure out the N that you need to take it on to a bigger study. So there's a lot of lovely tools out there that can really help you determine your power for your studies. So now my colleague will come over and really hone in on the experimental designs and case studies that have that we've seen in the past that have not worked out so well. So Sam?
Good morning, everyone. So the purpose here is really to kind of get you to think about the way you're designing your experiments. So we've put some example case studies together. They may be real, they may be not. I'm not going to tell you. But I'd really like you to think about these critically and shout out answers. I think we may actually have microphones as well if you'd like to get involved. But really, try and think about these things and then come up with your own solutions, and then we'll go through a few of our own. So study one we have here. We have some cute little mice. Researchers are performing untargeted metabolomics to study the effects of the gut microflora on the metabolome. They're using mice, obviously, and the plasma was collected at the same time by bleeding them into EDTA tubes. 50 samples were all collected in the same sample tubes, spun down, and the supernatant was aliquoted and frozen at -80. And the samples were inspected when they were received, and the samples were inspected upon receipt, and this is what they looked like. So I want you to take a second here to observe these tubes. This is just 3 sample tubes of what was received. Are there any potential issues that you can see with this experiment? Does anybody have an idea? Yeah?
Differences in the headspace and the tubes between them.
The headspace, definitely. Anything else you can think of? Anyone? Of course, I'm looking directly back at you because you were the first one to answer.
Hemolysis?
Yes, hemolysis. So based on those 2 observations, can you salvage these samples? Is there anything you can do to continue the experiment? So some suggestions, if no one has an answer for me. You could do a small pilot experiment on a few of the samples and see what the differences actually are. You could also potentially adjust the sample size to account for that difference in volume. you could include that degree of hemolysis in your statistical models. And you could also maybe just use only the red ones, the ones that have quite a bit of hemolysis. And then there is also the option to compare the hemolyzed versus the non-hemolyzed and subtract that signal out. And so some results from a study like this, and you can see the Venn diagram here. This is from untargeted mass spectrometry metabolomics. And so you see the hemolyzed overlapped with the non-hemolyzed and the partially hemolyzed. And you can see there are quite a bit of differences and some unique features that are observed between the three different types of samples. And so this take-home message here is that you need to establish clear guidelines and protocols for sample collection so that you can avoid these things in the first place. But if these things are out of your control, then there are different ways you can actually account for this and keep your study going. So now we have a study 2, which is a multi-center study. We have a study with 1,000 samples from TB patients and healthy controls also. And this was a 1-year experiment, and it was from 2 different collection centers. Everyone was from the same age range and the same ethnic background, so that was very well controlled for. And the goal of the study was to determine the difference in the plasma metabolome between the TB patients and the healthy controls. The control samples were collected at one of the institutes involved in the study, and the TB patient samples were actually collected at another center. They were all collected and shipped on dry ice, so everything was controlled for and sent to the University of Colorado. Yay! And upon receipt, it was observed that these samples were collected in 2 different types of tubes, and that's shown here. There was no additional documentation received from the centers related to sample collection. So are there any potential issues you can see with this study? And could this be salvaged? So once again, we have things that we can control for, so we have to figure out whether or not we can continue with this experiment. And so I'm going to continue on to the next slide, unless anyone has like an idea of potential issues. So we did some initial analysis with the 2 different sample tube types and found that with 3 replicate injections of each type of sample, we have some major differences. And you can see on this chromatogram around 2.4 minutes, you have a major peak right there, and it's not in manufacturer 2. You also have several other peaks that are showing up in the chromatography that are not observed in the other chromatogram. Oops, went the wrong way, sorry. So taking this into mind, you need to ensure before something like this happens that you have plenty of consumables. So if you're going to be collecting blood or plasma or any type of biofluid from multiple centers, that you should probably be the one that's providing them with their tubes, all of their consumables, preferably from the same lot, from the same manufacturer, and that you control for this at the beginning of your design. Let's move on to study 3, which deals with technician issues. So a lot of us actually have several people working in our labs, and they don't always do the same extractions. So we don't have one person that is solely in charge of extractions, except for maybe in our lab, but that's a different story. So we have a team of 5 graduate students. They're all trained, well trained at that, by the postdoc in the lab to perform a liquid-liquid extraction, followed with some SPE for plasma. And they've all done it a number of times, so it should be second nature to them. So they got a new grant, and one of the graduate students was selected to perform the sample prep on a large number of samples. It was going to take a long time, essentially, and on the 5th day, Billy got sick. He couldn't come to work, so Sarah, who takes good notes, and Marley, who doesn't, were assigned to complete the prep. I can see some problems with this one already. So both of them have their sets of calibrated pipettes, and all the samples were randomized so that there would be minimal, I guess, differences between the 2. But I see some issues. Does anyone else see any issues with the 2 new experimenters that are going to finish this prep? Anyone? No? Okay. So here's some results from this study. And we have a Venn diagram that overlaps Marley's results, Sarah's results, and Billy's results. Now take in mind that Billy was the original one to complete this this prep, but then the two other girls took over. And as you can see, A, which indicates Marley's results, she has 543 unique compounds showing up in her metabolomics study. So I can see that that's going to be an issue to continue this, or to even use her samples. What could have possibly happened that her samples had so many more unique compounds? Any ideas? Okay. So let's go to the answer, since we don't have one. So it seems that Sarah and Billy, they actually did their pipetting properly, per the protocol that was established in the lab. While Marley, and this is obviously just an assumption based on like a fake experiment that we've made up, but Marley seems to have pipetted the entire supernatant rather than just the portion that was required for the experiment, and it resulted in over 500 more metabolites. So the question is, what can we do to minimize operator differences in the future? And typically, it's having standard operating procedures, which all of us understand help minimize these problems, but also documentation so that we're all performing the same procedure the same way. So let's move on to study 4. So this is for those of you who are not working with biofluids, that you might be working with plant matter. We have a study that's comparing organic versus non-organic strawberries. I don't know if they got them at Whole Foods or not, but this is just part of the study. Samples were collected at 14 different sites, 7 organic farms and 7 traditional farms. They were analyzed fresh, and the prep was done, and the instrument analysis as soon as they arrived in the lab. Unfortunately, they didn't all arrive in the lab at the same time, so they were prepped and analyzed over the course of 3 days. So what do you notice about this? Actually, I'm going to go ahead and show this now. So they were prepped and analyzed over 3 days. Instrument analysis was done, and then we did a PCA on the resulting data. And can you see anything here, based on the day that they were prepped and analyzed, that would be a concern for any project? Batch effects. So you can see on the very left, you have your day 1 samples. They cluster pretty nicely, and the colors do indicate the collection site. So you can see based on the different colors that most of the collection sites do cluster within each other. But then you have day 2, which is way far over there on the right, followed by day 3. So you have some pretty severe batch effects that you're observing here in this study. And that can be a concern, especially if you're trying to do a long-term study like this where you're not receiving samples all at one time. So this is actually just a 3D view of it. It might even be a little bit harder to see what's going on, but there's even more separation there based on the day that they were collected and analyzed. And so this is a big issue with a lot of people doing long-term studies. The biological variation between samples can often be very subtle. And so batch effects can mask this subtlety. And so we have to minimize these differences due to external factors like extractor, day of extraction, instrument issues, because obviously our instruments don't perform the same way every single day for 365 days of the year. And we also need to try and randomize whenever possible to keep other variables as consistent as possible. So here's a little summary of the experimental design section. Your design can actually make or break your study, obviously. And SOPs are necessary for your sample preparation all the way to your data analysis. So everything that goes into a metabolomics study needs to have an SOP, a separate one, detailed, so that anyone that comes into the lab could do this experiment without much training. Proper quality control for quality assurance ensures your credibility in experimental results and helps you get published if you have some QC. And then you want to plan out an experiment in detail before sample collection so that you're preparing for any potential issues that might arise, like tubes or people that can't complete their extractions. And lastly, of course, you want to document everything. Lab notebooks, OneNote, whatever you do, document it all. All right, so that concludes the first section. So I will hand it over to Dan.